This video is meant as an introductory video to students just starting out in Botany and Dendrology classes, especially those who are taking an online version of either one of these classes, because they tend to not get as much opportunity to study with a skilled professor on how to use a dichotomous key and identify plants. When you're just starting out, looking at a dichotomous key or trying to use one for the first time can be extremely intimidating, especially because when you read the questions, you won't understand half the words. I hope to demonstrate to students how easy you can use a dichotomous key and at times the patience that is required to use a dichotomous key. A lot of times new students will think that the dichotomous key is too hard of a way of identifying plants and that it's much more easier to just go buy a book that has a bunch of pictures in it, and especially one of those books that has flowers categorized by color, and just going to open up to the pink flower section and I'm going to flip through the pages and I'm going to find the rose that I'm looking at. Yes, you can do that, but if you're skilled at a dichotomous key, it's often quicker to use that once you're skilled at the key, because flipping through a color book, you'll flip and flip and flip and flip and flip and flip and flip, and maybe you'll miss it, and then you'll go to the end of the section and you'll have to go back and flip and flip and flip and flip, and then you'll find a picture that you're pretty sure is your plant, but only because the pictures kind of look like it, but it's actually another plant. Because there's, especially as the example previously given in the rose family, it's very difficult to tell the difference between Gymnocorpa or Nuctana, especially by pictures. The flowers will look very similar, and you really have to start looking at like the armor, the thorns, to see the difference. So pictures aren't always are rarely the most effective way. It's actually much more easier to use a dichotomous key to be more accurate. It's much more easier to be more accurate. But picture books are still very valuable as a reference once you've used the dichotomous key and feel like you've successfully come to an identification. If your book doesn't have a picture and only a verbal description, which can be extremely accurate, it's nice to be able to have that other reference book to open up to the pictures and go, oh yes, this might actually be for sure what I'm looking at. I'm pretty confident that this is what I'm looking at. So jargon is super important to understand going into any dichotomous key, and it's a slow process when you're just starting out, because you're not going to know all the key terms. So some learning families can also be extremely valuable, because it does require a lot of upfront time to study and learn the families in your area. You don't have to know all 350 or 450 families. You just have to learn the families that are most common in your area. There's going to be like 50 or 60 common families, and if you can learn those families, then that cuts your keying time sometimes more than in half. Flashcards are almost essential in my mind to learning families or individual species. I like to use the larger flashcards and print out color pictures. I did so in the library at the college, and it's a small fee to print out color photos. Another great way for any class, if your teacher happens to have PowerPoints, you can also print out your PowerPoints into squares and then just cut them out as flashcards. There are many great online resources. If you're just going to have to search for a couple of them, and there's some pretty great apps that you can use like on your phone or tablet. iNaturalist, I think, is probably the best. I wouldn't recommend any of those apps that claim that they can identify a plant or a tree through a picture. They just don't work at all, in my opinion. But with iNaturalist and other social network identification apps, those are pretty neat, because at the end there's another enthusiast or high-level professor of some kind that is looking at your picture and helping you identify it. The great thing about learning about plants and trees and being able to identify them is you train your brain to see things that you just never noticed before. And you start respecting these plants you start seeing these plants in a different light. And all of a sudden, a walk through the forest that might have been more just a quick jaunt becomes a much more enriching experience as you begin to notice a whole plethora of living organisms that you just never really fully seen before. So let's get to it. We're going to go up to North Umpqua. We're going to try to identify a bush, a tree, some flowers, and I want to end with giving you an example of using a laboratory setting to identify plants. I've been walking around for a while and been noticing a little bush on the ground that I think will be a really great place to start our keying practice. I think that bushes and trees can be a lot easier to key out, to use a dichotomous key on, because their features tend to be a lot more prominent than a small flowers is. And so I think it's a good place to start for some good practice keying. This particular grouping of bushes, we're pretty lucky because we happen to found some that still have some berries on them. This one in particular, lots of red berries. And that's going to be really important to help us identify what this plant is. Just trying to identify something out of the twigs and the leaves leaves some room for uncertainty and ambiguity, and if we happen to have prominent features like berries and flowers, that can help us be more confident with what we feel like it is. This is the book we'll be using to key out this particular bush, Manual of Oregon Trees and Shrubs. Additionally, I have Trees and Shrubs of the Pacific Northwest with us. This is a great book that I'm going to use as a companion book to anything that I key out in this one, or another book that we'll be using for flowers. This one happens to have some really beautiful pictures that can help us be more confident with our decisions when we come up with what we feel like the plant is. I also have a little notebook here that I'll be taking some notes with. Turning to the page that begins with the key for shrubs. It's labeled Key to the Pacific Northwest Broadleaf Trees and Shrubs. Most leaves reduced to sharp spines. Few leaves have normal blades. Or most leaves not reduced to spines. Most leaves have normal wide blades. Well, there definitely doesn't seem to be a lot of sharp spines on this. And the leaves seem to be normal. Second question, so it tells us to go on to set question 2. Leaves simple or leaves compound? I think that you can kind of have an argument that these kind of look like an Oregon grape and they may be somewhat of a compound leaf, but I'm going to say that they're simple leaves. Leaves and buds opposite, leaves and buds alternate, or leaves and buds alternate. Definitely alternate. And that's going to have us go on to question 15. So, question 15. Leaves palmately lobed and vein and maple-like, or leaves not palmately lobed and vein? Well, they are definitely not maple-like, so they are definitely not palmately lobed and veined. So question 20. Branches armed with thorns, buds red, fruit in a brightly colored pone. Well, our fruits are definitely brightly colored, but our branches armed with thorns. There are not thorns. So we're going to go on to the second half of that question, branches not armed. So that's going to have us go on to 21. Leaves have 2 to 4 distinct glands, and petioles or base of leaf, stipules common. Distinct glands on petiole or base of leaf. This is like a little bump or I don't know, nub or something at the base, and it can be somewhat hard to tell or see. And I never really liked that question when they're asking for glands. If you see little dots or little bumps, that's a gland. So no glands on petiole or of the leaf, or on the leaf. 22. Leaves wedge-shaped, apex much wider than base, apex 3-lobed, tend to persist. Yeah, leaves wedge-shaped, or leaves not wedge-shaped, apex not three-lobed, and may be deciduous or persistent. I'm going with that one. Leaves not wedge-shaped. 24. Leaves three-veined from the base of the blade. Petiole short and round. So they are not three-veined from the base of the blade. Leaves not usually three-veined. So that would be 25. F three-veined, then the petiole is distinctly flattened. So it is the petiole distinctly flattened. Yeah, I mean it seems flat. I don't know if it's distinctly flat, but it seems flat. So let's go on to 25. Leaves linear, long, and slender. Those are not that long and slender. Leaves not linear. So, question 26. Leaves persistent, stiff, thick, leathery. Previous year's ivies present with current leaves. Leaves not deciduous, not as above. So these are definitely not stiff and thick, which means that they're going to be a persistent, more year-round like leaf. So these are more of a deciduous leaf. So question 45. Jumping ahead. Young twigs distinct, ribbed or ridged, but not triangular, and bright, rich, Kelly green. I'm not sure what Kelly green is. But these young twigs distinctly ribbed or ridged. They definitely have a distinct rib to them, but they are fairly round, so they're not triangular, and they are a bright, rich green. I don't know about a Kelly green. I'm not sure about that. So that's going to have us going to 46. Leaves both simple and trifoliate. So we have all simple leaves, no trifoliate. Small, rich green twigs, bright green, and ribbed. So yeah, we already know that the twigs are somewhat green and ribbed, but they're definitely not simple and trifoliate leaves on the same plant. All leaves simple. Definitely we have all leaves simple. Non-trifoliate. Elliptical, ovate, or oblong are leaves. So our leaves, let me get a good example. Yeah, our leaves are definitely more elliptical or ovate or oblong. And buds red. Let's see if our buds are red. Yeah, this is pretty awesome. Our buds are definitely, trying to pick something that's not going to hurt the plant too much. But these buds are definitely red. I'll pick some pictures of it later. And that means that this key is saying it's Vaccinium parvifolium. Page 243. Let's see if that description matches what we've keyed it out to. Vaccinium parvifolium, red huckleberry. Well, this seems pretty good. This picture seems pretty awesome. Okay, this picture sucks. It's just hand-drawn picture. But red, rigid stems, red berries, very thin leaves. These are very thin, red berries. And if you read the description, which I'm going to save us for some time and not read the description, this is what we have. This is a Vaccinium parvifolium. This is a red huckleberry. We'll go find another plant to key out. Keying out that bush went pretty well. So let's try a tree. So with this tree, I'm going to do something a little differently. So instead of reading the entire question verbatimly, I'm going to only say the key words that are standing out to me in this key and show you how quick it can go. Question 1. Leaves scale-like, all-like, or both, or leaves not as above. Not as above. Leaves are definitely not scale-like or all-like. Go to question 7. Yeah, question Sheffer. Most or all needles born in clusters? No, all needles appear to be born individually, connected to the twig separately from each other. All needles appear, yeah, they're definitely appear to be born individually. Definitely born individually. So question 10. Needles deciduous or needles evergreen? We know it's an evergreen. Number 11. Single needles born in distinct woody pegs. Pegs may be rounded, squared off, needles not turning, needles not as above. So definitely we have some woody pegs. woody pegs easily seen, woody pegs less conspicuous, or are they less conspicuous? So I think that they're fairly harder to see, so I think they're less conspicuous. So let's continue reading that question. Rounded, point forward on the twig, needles under 3/4 long on blunt. So yeah, they definitely appear to kind of be more rounded and pointing forward. So that gives us the genus Tsuga, or hemlock. And there'll be more questions to answer. So question 1, leaves flat, varying in length, tend to be 2-ranked, or leaves thickened, not 2-ranked. So 2-ranked, in this particular case, is meaning that they have 2 different sizes. But they're definitely have two different rank sizes, which gives us the heterophylla, which is what we have, Tsuga heterophylla. But with the bush we did previously, it happened to just give us the answer directly. It just said, here's a bunch of questions, and if you answered no to all these and yes to these, then this is definitely what you have. You definitely have this genus and this species. When we do flowers, is we answer a bunch of questions, you'll get the family. Cool. We answer a bunch more questions, we get a little closer to what it might be, by also getting the genus. And then finally, we have to answer some more questions to get the exact species. So I found this nice little botanical corner here. We got some lilies, we got some mints, we got some scroffs. I'm going to try to use our handbook of northwestern plants to key out this lily. I already tried and I got to an answer that I'm moderately comfortable with, though I don't know for sure that it's the right answer. When we're dealing with flowers, their parts are so much smaller, and the jargon used to try to describe these parts can be a difficult challenge, especially if you don't put any time into learning these words. So it's very important to learn the botanical language so that when you read words like calyx or subtended or sessile, you know what they're referring to without having to look up the glossary. Regardless, you still are going to constantly be running into terms that you're going to need to use the glossary on, because there are so many botanical words. It could be challenging to know them all. So I'm going to skip right to the lily because I know it's within the traditional lily family. So that's where an advantage of putting in the time of knowing the families will get you. I shave off a huge chunk of keen that I don't have to do. And I can go right to what the family is. There's a little purple lily here. Is the guy we're going to try to key out. They opened up one of these lilies to dissect it, to try to answer some of these questions. Styles separate. This is where I kind of messed up on earlier, because I kept thinking style as in the entire pistil, or rather where the style is just a part of the pistil. You have the ovary, the style is the tube, and the stigma. The style is the tube of the entire pistil, and that's where I messed up on earlier. The style is united. The tube is completely united all the way up. So style is united to the middle or above. So yes, down to question 7a from 1b. Sepals and petals distinctly different. Again, terminology, sepals, petals, you should know some of these things. If you don't, you need to study that. But you can see that the entire flower, well, maybe you can't see, But the entire flower is of one color, and it's tough to distinguish the difference between where the petiole or sepals begin. And the petioles and sepals are distinctly different, so they are not distinctly different. So let's go to question 7b. Sepals and petioles quite similar, all petiole-like, which is what we have. They're all. So then flowers small, many in scapiose, bracted umbel, or a bracted subcapitated cluster. I know what an umbel is. I'm not sure about a subcapitated cluster, but I'm not going to look it up because we definitely have an umbel of flowers here. The other question would have been flowers not scapous, bracted umbels, nor a bracted subcapitated cluster. Subcapitated. Scapose, I am going to look that up real fast because I think I know what it is, and before I say it, I'm going to look it up. Scapose, with the flowers borne on a scape. Well, duh. Scape, a leafless peduncle arising from the ground. Okay, so that is what I thought scape was, and that's what we have. I don't want to pull out too many of these guys, but this thing has no leaves along this big long stem. It's a big old long stem. The leaves are at the base, which is basal. So the leaves are basal, which I do believe will become a question. That gives us to question 11a or 11b. So 11a is plant with an onion or garlic odor, perianth parts free or nearly so, and in question 11b, plant without an onion odor, perianth parts united below into a tube. So before we get to the part where they're talking about perianth parts united, let's just see if we can smell an onion or garlic. Onion or garlic odor. And I even tasted a little bit earlier. See if I can see if there was an onion taste, and not recommended because there could be like allergies and stuff which you don't know you have to these wildflowers, but you know, I live on the edge like that. Anyway, onion, there was plant without an onion odor, definitely no onion odor. Plant 11b, plant without an onion odor, and perianth parts united below into a tube. A perianth, look at this, when I took this dissecting apart, when I dissected this flower, we could see that the perianth parts, being the petioles and the sepals, are united into a tube. Perianth parts united below into a tube. That is correct. We can be confident we can move forward. 11a, fertile stamens 6 or fertile stamens 3. There's 3 definitive stamens in the one I dissected. And we know that they're fertile in the fact that we can see a bunch of pollen, which is going to appear to be yellow, on the stamens. So we got a lot of yellow pollen, powdery substance. And this is where having a monocular can be, a monocle can be pretty advantageous to helping you see these things. But I'm going to go with fertile stamens of 3, not 6. 13a and 13b are our next two questions. 13a, inflorescence in open umbel. Not fully sure what an open umbel is. I'm gonna guess it means that the individual flowers are kind of growing apart from each other and not so connected as they are here. They're pretty much all one glob at the base. So I'm going to say that that's not an open umbel. This is clearly very dense, and that is what the next question 13b says. In inflorescence, a dense umbel. That gives us our genus Dichotomous. Alright. And in this particular book, and that we don't have to do any more keying for that genus, because this book only has one species of that genus, which is Dichotomous congestum, or common for field cluster lily. I'm going to read this to see if this sounds like what we have. Scapus, 60 centimeters tall or more, sometimes bent or twisted. So we definitely have bent and twisted inflorescences. And this particular one looks like it's around 30 centimeters. So it's within our height tolerance. Flowers in compact cluster. Yes. Few 20 flowered. Don't see any that are 20 flowered. They all look to be more like 6 flowered, but I've seen these where they've had a lot more flowers in their cluster. Subtended by several ovate bracts. Subtended means immediately below and attached to. And they are like these are somewhat ovate, which means kind of round or egg-shaped. And there are somewhat ovate bracts that are subtended to the umbel cluster, or the umbel of flowers. Flowers light to dark purplish blue. So we definitely have some light to dark purplish blue flowers. 12 to 20 millimeters long. Stamens with anthers 3. Yeah, we already said yes to that earlier in the key. Short filaments united to the perianth, deeply lobed in staminoidea 3. So our that sounds somewhat correct. We have short. Our stamens are very short and are united to the perianth, and they are 3 of those. But I'm not sure what a staminoidea is. Staminoid, a modified or abrupt stamen with which does not produce pollen, which is plural staminoidea. Maybe earlier I didn't really see anything when that was in the key when they were asking if there was any stamens that weren't fertile, but perhaps these little purpley looking things, well, the whole thing's purple, but there's this little bracky, I don't know if that's the right word either, obviously not, but I don't know, there's this little thing sticking up in between the petiole and the stamens that's definitely not stamen. And perhaps that is what genetically in the past could have been another stamen, which is now a staminoidea. So I'll just say yes to that and say that there's a lot of yeses in this description, that I can be reasonably confident that even without a picture, this plant is the Liliaceae Diclostemia congestum. We'll look that up on the internet later on. We already practiced keying out some plants out in the field. But sometimes plants can be very difficult to identify in the wild. They'll have extremely small features. They require more sophisticated methods for identifying them, which is why we're here in the laboratory. Try to show you what it's like to identify and use the tools in a laboratory setting. Again, we'll be using the handbook of Northwestern Plants by Botany and Dendrology. We start out with separating the keys into either monocots or dicots. One feature that we'll be using to determine that this is a dicot is the vein pattern. Unlike the lily we identified earlier, which has parallel veins in the leaves, this veins are splitting in part, which they will call net veined. So these is a dicot, so we will start keying from there, or start at the dicot key. 1A, petals none, or 1B, petals present. Well, we have petals, so we'll go to 1B. Next question, 42A. Petals free from each other or only slightly joined at the base. Or 42B, way back here, says petals united at least at the base. So petals pretty united here. They're joined, these petals, that lobe out here plus this foot of the petal, are joined at the base. So that means we go to 42B. So 107A, stamens more than 5. Or 107B, right there, stamens 5 or less. So let's check that out. Ooh, we're gonna have to zoom in. So with our microscope and using some tools to get in there, we can start to answer that question. You can see that we have only 4 stamens. So that allows us to know that we need to go to continue on with 107B, stamens 5 or less. So 114A, ovary inferior, or 114B, over superior. So let's see if we can't get in there. Look at the ovary. Anyway, we have gotten the calyx out, open, and inside the calyx, not below it, we have our nutlets, that would be the swollen part of the ovaries. And so, yes, ovary are superior. 123A, corolla more or less irregular, or 123B, corolla regular. Strongly two-parted flower. It is definitely not regular. So it is more or less irregular. So 123A to 124A. Leaves alternate or basal, or only the lower leaves opposite. Or 124B, leaves all opposite or whorl. We definitely don't have a bunch of leaves down at the base, or only the lower leaves opposite. All the leaves are opposite, so leaves opposite or whorled. And they're kind of whorled. You can see that we have opposite leaves and then rotating, rolling around, just kind of rotating around. We have 2 more leaves opposite here. 127A, ovary deeply four-lobed, separating into 4 nutlets, style usually arising from the base of the lobe, stem square in cross-section. We can already see right here in the microscope that we have 4 nutlets. It might be hard to see all 4 of them. So 1, 2, 3, 4 nutlets. So that looks promising. But what about the square cross-section? It's square enough. I think that if I was just starting out, I wouldn't call that square, because it's kind of convex and concaved, and it's not what I would consider square. But those regardless, let's go ahead. Well, let's just go ahead and read this 127B. Ovary not deeply forelobed. Well, stop. We don't have to go any further. Ovary definitely forelobed. F separating two nutlets, doing so only at the maturity. So we definitely have ovary deeply forelobed, separating into 4 nutlets. So we don't have to go any further. So that gives us 2, yay! Lamiaceae, which is the mint family, which is what this is, the mint. Now that we have a family, we can start trying to figure out the genus. Ovary merely forelobed, nutlets attached by inner margins. Or ovary deeply foreparted, nutlets attached by their bases. Alrighty. So our main question here, nutlets attached by inner margins or attached by their bases? Well, that's going to be difficult. It's a good question, and one that I'm glad we have a microscope to use. And technically you shouldn't have to use a microscope. These books are designed to where if you had a 10 by monocular hanging around your neck or something, you can use that. But you can see when I touch these, hopefully you can see, pretend like you can see. When I touch these nutlets, they really just want to fall apart from each other pretty easily, only attached at the base. Cool, we get to move on to 2A or 2B. Corolla scarcely two-lipped, the lobes nearly equal, or corolla usually four-lobed. Or 2B, corolla distinctly two-lipped, the lobes unequal. I think you can remember from before that we have a huge distinct two-lipped corolla. Just 2 massive different sections there. So 2B. Moving on to 4A. Fertile stamens 2, fertile stamens 4. Now again, I mentioned this earlier. The fertile stamen will have pollen on it. You can see that it has a little yellow, powdery substance. So again, this is where a microscope can be handy to really look at whether or not these stamens have yellow pollen or some type of pollen on them. And they do. Fertile stamens 4. Calyx teeth equal or nearly so. Calyx teeth equal or nearly so. Calyx two-lipped or teeth clearly unequal. Sorry I say so like every freaking minute. Alright. So we can see the lower part of this has 2 really sharp sepals, and these upper sepals have been fused into a 3 little section part. Or question, calyx teeth equal or nearly so? These are definitely not equal. The sepals have 2 distinct different patterns. Calyx two-lipped, or teeth clearly unequal. Definitely two-lipped, and teeth are clearly unequal. 9A. The 2 calyx lips entire, calyx crested on upper side. Or the 2B, the calyx lips 2 to 3 tooth or lobed, calyx not crested. So let's look up what the word crested means. Crest, a ridge or projection. So these questions to me are a little ambiguous, and it would be difficult to decipher the difference of where I would need to go between 9A or 9B. The 2 calyx lips entire. These lips look entire to me. They look smooth. But perhaps they're going to say that these little individual projections up here prevent it from being entire. Calyx crested on top. So again, crested is a ridge or projection. So yeah, they have a projection on top. on upper side. And the other 9B, calyx lip 2 to 3 tooth, or lobed, calyx not crested. So to me, I would almost want to go 9A. So let's just go ahead and look at 9A. I know it's not 9A, but let's go ahead and check it out. Alright, when you read the description, so 9A, by the way, gives us to Squatillaria. So when you read that, it almost sounds like what we have. I think that this general description of the genus still could fit our plant. But when you get down to the individual species that are in this book, which are 3, you don't really key out to any one of those 3 in these little key here. And if you read the individual descriptions of those species, it doesn't quite sound like what we have. So let's go back to that question and go ahead and move forward to 9B. The calyx lips 2 to 3 tooth or lobed, calyx not crested. So again, I feel like we'd say that this thing's crested, but we definitely have 2 to 3 tooth or lobed if we're considering these little things up here to be our tooth or lobes. And so in 10A, inflorescence dense, terminal, or inflorescence axillary, or sometimes also terminal. So terminal is like just at the end. So they're saying, is the inflorescence dense at the end? So at the end of this, we could see that there used to be a bunch of flowers. And this is definitely dense at the top, at the tip of this flower. If you get closer and look a little closer, you can see that these flowers do get a little closer to each other as it gets closer to the terminal, to the end. So let's go ahead and agree with that statement, which gives us to prunella. Flip into page 3. We are in luck. There is only one species of prunella in this book, so we don't have to continue to do any more keying or dissecting to figure out what we have. This happens to have a nice picture right there in the book, which this book doesn't have very many pictures, but this looks exactly like what we have, and if you read it, it is what we have, which is Prunella vulgaris. Heal all or self-heal is the common, so that sounds like a pretty sweet plant to know.